Hello and welcome to Poulter's Pod. I'm going to give you now a little Year 10 biology separate science lesson. I think we're going to focus, since we've finished the organisation topic and we've got a bit of time, I think we'll focus on sort of recapping and a bit of stretch and challenge as well for these sort of these 2 hours we've got together. So just to start off with, recapping our current knowledge of enzymes. What do we currently remember? So hopefully you already have a bit of a strong working knowledge of the kind of the basic structure of enzymes, how enzymes work, factors that affect their action, and how they're used in digestion. And the starter in this first lesson is you guys using your current working knowledge, maybe your revision guides if you can't remember everything, to kind of recap those digestive enzymes. I'm not going to focus too much more in this first sort of section on that. I'm more interested at the moment understanding how our actual enzymes work and the factors that affect them. And then the second lesson, in my second video that I'm going to present to you, we'll look at kind of more that stretch and challenge and the kind of experimental application of enzymes and how we can investigate their action better. So hopefully you remember that enzymes are biological catalysts. Well, their biological bit comes from the idea that they're made of protein. The catalyst part comes from the fact that they speed up chemical reactions and they're not used up in the process. They can be used over and over again. And if you remember in my lesson, I taught you that actually enzymes can speed up chemical reactions something like 14 million times faster. So they're really essential to life, because without them, all the chemical reactions that occur in our cells, like respiration, if you're a plant, photosynthesis, the digestion of foods, all these sort of metabolic chemical reactions would be too slow for our cells to function correctly and they would die. So they're really essential. But how do they do it? How do they work? So I'm going to draw for you a really kind of simple kind of cartoon of an enzyme. In reality, they don't really look like this, but it works for our purposes at GCSE. So here's my kind of basic enzyme shape, a little bit like a Pac-Man, if you like. Remember, that's a protein-based molecule. And then we have the molecule that fits into it. And hopefully you remember that's called the substrate. So the substrate has a shape that is complementary to that of the enzyme. Okay? It should fit in together like a lock and key. The most important part of the enzyme there is the active site, okay, which has a specific complementary shape to our enzyme. When the enzyme and the substrate are joined together, I'm going to draw that now, something like that, we form what's called an enzyme substrate complex, which is a really key bit of terminology you want to try and use in your exam questions. And what that basically means is that our substrate is collided, it's fitted within the active site, and therefore the enzyme can then catalyse a reaction, which might be breaking down that substrate or actually joining substrates together. And eventually we end up with our enzyme again, and our sort of our substrate with our product has been formed. In this little example here, I'm going to just pretend that it's broken down into two separate parts. So something maybe like an example might be amylase breaking down starch into things like glucose and so forth. All right, so that gives you a basic kind of overview of the structure and function of our enzymes. Now I want to sort of talk about factors that could affect their action. So there are two main factors that can affect the action of our enzymes. One is temperature and the other is pH. They affect the enzymes in different sort of ways, but before we kind of delve into why each of those factors affects enzyme action, we need to consider what needs to happen for the enzyme to actually catalyse the reaction. Well, first and foremost, we need a collision between the enzyme and its substrate. The substrate can't just collide anywhere on the enzyme, it has to collide specifically with the active site in order for that reaction to take place. So we need those kind of collisions going on. So kinetic energy there is obviously very important, because kinetic energy will determine how much those particles are moving and the kind of frequency we get with collision between the enzyme's active site and substrate. So the more collisions I get, the more enzyme substrate complexes I get, therefore the faster the reaction is going to be. On the other hand, if I change the shape of my active site to a way that makes it no longer complementary to the substrate, the enzyme can no longer catalyse a reaction. And we call this denaturation, or the enzyme is denatured, however you want to kind of word it. And both temperature and pH can cause denaturation of enzymes. So the first thing we want to consider is the effect of temperature. So with your typical graph, what you'd expect to see, temperature on the x-axis, and then enzyme activity or something like that on the y-axis. And what you'd expect to see is, as temperature increases, the rate of reaction increases up to a certain point where we reach kind of the peak, call that the optimum temperature, and then very quickly the rate of enzyme activity starts to decrease and eventually actually goes down to 0. So that's kind of descriptive for our pattern, and the optimum temperature will be in different places depending on the type of enzyme. All the enzymes in my body, their optimum temperature should be around 37 degrees C, which is my body temperature. But if I'm a plant, it's going to be considerably lower, maybe around about 20-25 degrees C. So bear that one in mind. And as temperature increases, the amount of kinetic energy in the system also increases. So therefore, I'm more likely to get collisions between my enzyme and the active site. Okay? Now, the more frequently that happens, the more enzyme substrate complexes I form, the faster the reaction is, to the point where I reach the kind of optimum point where those collisions and enzyme substrate complexes are forming. But once I reach a kind of a threshold temperature, I go beyond that optimum, the enzyme begins to denature. So the bonds that are holding together the enzyme and giving it that very specific shape are being broken, and therefore the enzyme's active site shape changes, and as a result that substrate can no longer fit in there correctly. It's denatured, and that explains that kind of drop off. With pH, on the other hand, it's a little bit more complicated than that. At GCSE it's kind of difficult to explain. You can generally keep it quite simple. What I would say for GCSE knowledge is knowing that each enzyme has a specific optimum pH, and if you think about your digestive system, there's variation in your pH. So in your mouth, for example, it's going to be around a little bit above 7 probably. In your stomach, it's going to be pH 2. In your small intestines, it's going to be pH 8. And different enzymes have specific optimum pHs. So amylase in my saliva, for example, can function perfectly well there, but as soon as it reaches my stomach when I swallow, the pH is far too low for it, actually cause it to denature, so I can no longer actually digest starch in my stomach. It's all done in the mouth and also in the small intestine, but of course I have to add more amylase in from the pancreas or the actual cells lining the small intestine. Once you start getting to the extremes in pH, too high, too alkali, or too low, too acidic, the enzyme will denature. And that's generally speaking okay for our purposes at GCSE. So there's a kind of a little brief overview there on the enzyme action. I've got various other activities and questions in the lesson that I'd like you to have a look at, make notes, etc. They're going to help you out. Okay, so this is Dr. P signing off. Have a good one. Cheers.